The next speaker is Professor Anja Kipar from the University of Zurich, Switzerland, and the title of her talk is Feline Coronavirus Pathology.
So first of all, thanks to the organizers for inviting me. It's sort of a bit of a home play because, as you can see here, this is not a disclaimer, but I'm currently director of the Institute of Veterinary Pathology at the University of Zurich, but I was a professor of veterinary pathology at the University of Liverpool, and I've been working on a different type of coronavirus since the pandemic, and that's, of course, SARS-CoV-2 with the molecular biologists like James Stewart and also with Julian for quite a few years. And now I'm coming back to where I started, more or less, because I did my habilitation, if that means anything for you, outside the German-speaking countries on the pathogenesis of FIP. And yes, I'm also still affiliated with the University of Helsinki. So the title I was given was Feline Coronavirus Pathology, and that's what I want to talk about today. So brief outline, I'll speak a bit about the history, then the virus, and my disclaimer is not anything commercial. My disclaimer is, virologists, please don't be disappointed because my virology will be very limited that I bring. Clinicians, please don't be disappointed because I'm not talking about clinics. Pathologists, you will not be disappointed. Because that's what I am, and it's not as exciting as the outbreak was, but I think I can set the ground so you understand about what feline coronavirus does and why I tend not to agree straight away that FIP is a model for any human coronavirus infection known so far. And I would say luckily. So brief history. So in 1966, the FIP was named and thoroughly described. That was in the States. And before that, in the 50s and 60s, there was a disease syndrome observed in the USA. But this is not an American disease, because in 1942, there was a report from Naples on the Peritonitis infettiva de gatto. And it really describes the same condition. So it must have been at least that's the earliest description of it. So then 1968, the viral etiology was demonstrated by Jerry Ward, who then turned, he's a pathologist, but a lab animal pathologist. But it was then him in 1970 who suspected coronavirus. And this is another name all the virologists know, Ab Osterhaus. Also, he did his PhD on feline coronavirus, on FIP. And he confirmed it was coronavirus. And there is the name Niels Petersen for the first time. I think he was also doing his PhD on the disease. So then Niels Petersen was able to propagate the virus in culture, one of the viruses. And Niels Petersen also wrote in one of his papers why he suspects FIP only emerged in the 20th century. And maybe he hypothesized it was because of a virus evolution of feline coronavirus alongside that one or those ones of pigs and dogs, and also that it had needed time to evolve from a solely enteric one at that stage. But also he mentioned that was the time when the keeping and breeding of cats changed a lot because they became really popular as pets and they introduced cat litters, etc. So maybe that helped with the scenario. So Severin Tasker was mentioned before, and I would like to alert you of the recent ABCD guidelines that came out in 2023, a really up-to-date review of FIP, including the infection, the pathogenesis, clinical features, and treatments, and a surprisingly unbiased one. I mean, it's tough to read, but it's extremely good, and a lot of my references here, or some of them, come from this paper. So a bit about the virus, just a few keywords. I said I'll keep it short. It's an alpha coronavirus, and the ones that have been everywhere recently and now the human ones, SARS-CoV-2 is beta coronavirus. So feline coronavirus is, of course, a single-stranded positive sense RNA virus. It has got about 11 putative open reading frames. And just, I show you this because some of these keywords come up later. So we have, of course, the replicase polyprotein when it's transcribed, and we have several structured proteins, the spike one, the membrane, the nuclear protein, the envelope protein, but we also have the non-structural accessory proteins, and they're listed 3A, 3B, 3C, 7A, and 7B, and they at least play some sort of role when it comes to development of disease or transmission, etc. So S is the one that otherwise is the most important because that induces the antibody response, and it comes to binding as well. So let's have a look at S briefly. It has subunits that, and that's for the binding and entry into the cell. We have a cleavage sites as well, and there is one mention already. There are some mutations that there was a big hype about that these mutations are the FIP mutations, and if you find them, that would mean a cat has FIP. One has to tone that down a little bit because it actually shows that the virus goes systemic, which anyway any coronavirus does in cats. But I'll come back to that in a minute. But it has been of relevance for diagnostic purposes. So there are two distinct serotypes, 1 and 2, and serotype 1 is the most prevalent worldwide. Unfortunately, it also is the one where no one knows the receptor of, despite trying for quite a while, and it's also the one that doesn't grow in culture so far when you want to just culture an ordinary. Whereas serotype 2, and so far it's a little bit, I think, as FCOV23, it's a recombination with a canine coronavirus, from which it has most of its S. That grows readily in culture, so it has been used for any in vitro work over decades. So there are these two pathotypes that are everywhere, and Niels Petersen introduced these in 2009, and he spoke about the ubiquitous enteric biotype as feline enteric coronavirus, and spoke about the FIP virus being the virulent biotype. I really like what Severin and colleagues have introduced now, because they cannot be discerned serologically or genetically, to be a bit provocative, and they suggest to call them the less virulent coronavirus and the FIP-associated one, because forget the enteric, the less virulent one is not just enteric. Yeah, so let's speak about the pathological features of coronavirus infection and FIP before I come to the pathogenesis. So before I forget, feline coronavirus only targets enterocytes, monocytes, and macrophages. There has been no evidence that there is any other target cell in the feline body. Yeah? So, insofar, it is very different to SARS-CoV-2 or other coronaviruses also in animals, because there are respiratory ones and enteric ones. There has not been any evidence that a respiratory epithelial cells or an alveolar epithelial cell can be infected by a feline coronavirus. We can probably manipulate them, but naturally, that's not the case. So the clinical disease, one can say, I mean, it was mentioned, it's recurrent fever, it's malaise, anorexia, weight loss, and basically why it's so variable is because the clinical signs reflect the distribution of the typical inflammatory processes. Like if we have the so-called wet form, we have the effusion. So I'll leave this to the clinical experts who will talk later. Yeah? This is more my field, so what are the gross findings? We have a fibrinous and granulomatous serositis. So we have protein-rich serous effusions. We have pyogranulomatous lesions in organs, and there are a few examples. That's top left is the kidney in these white foci. That's a typical inflammatory process. You can have a serositis like it shows on the bottom jejunum picture, where you have granulomas that really sit on the serosa, and you actually, even when you do your PM, you can't be sure that's FIP. Yeah? So then the word wet and dry FIP was mentioned. Basically, this is a clinical distension, because when you open up the cats It's extremely rare that you see what is on the bottom right, that you solely see effusion and no additional lesions. That would be a classical wet form, but they are usually mixed. So we have the granulomatous processes as well. So what are the histological findings? I spoke about the serological lesions, that's diffuse ones. And you see on the right, there is fibrin. And in the fibrin, you've got these small aggregates of macrophages. Yeah, and a general pathologist would not call this granuloma because a granuloma has a composition like Robert Koch and so described and Virchow for tuberculosis. But what it means, it's an infiltration that's mainly comprised of macrophages. They are the dominant cells. We have neutrophils in there, like it was mentioned before. And on the bottom right, you see abundant viral antigen there. So then we have a vasculitis that to specify is indeed a phlebitis, so an inflammation of a vein. And the very close picture of the kidney, you see this little white line, and that is such a granulomatous phlebitis, and it usually primarily affects the stellate veins that are just beneath the capsule. And you can see this here, and that's a phlebitis and periphlebitis with viral antigen. And when it comes to the brain form or the neurological form, do not get misled. There is no infection of the brain parenchyma. Feline coronavirus does not infect neurons. It's very different from what we see in our K18 mice that are SARS-CoV-2 infected, where we find the virus in almost every neuron. The brain form means we have granulomatous infiltrates and mainly a granulomatous phlebitis in the leptomeninx, and we can also have it in the choroid plexus. Nothing in the parenchyma, if then it stretches from the leptomeninx and goes a bit between with the gyri and sulci, between the structures of the brain. So the distribution of the lesions, yes, we did a big study and reported that the majority have the peritoneum affected with and without effusions, as you can see. The kidneys are the most frequently affected ones where we see the phlebitis, followed by the mediastinum, the mesentery, mediastinum, then the lymph nodes. Then when I said brain, as I said before, it's the leptomeninx, yeah? The eyes very often, but don't forget, in the eyes, the leptomeninx is just stretching from the brain towards the eyes, and that's why we see the brain involvement when it comes from the leptomeninx, and we can have a phlebitis mainly in the retinal vessels. But it's always seen, when you really check, it's always seen with brain lesions. So, for example, the lung can be affected, but the lung is affected by vasculitis, by the phlebitis, and it's not a respiratory infection, as I said. So the clinical course, I just about the time frame you're looking at. You can have animals that develop the wet form very rapid, but the dry form, it can be several weeks. And we initiated an FIP outbreak once in a sort of semi-experimental condition, and we introduced SPF cats to cats that shed the virus, and it took between 6 and 42 days that these animals, when they developed FIP, showed clinical signs or started to show that. And there were recurrent waves of clinical disease, and I think cats are really quite tough. They only show, you find clinical signs when they're almost dead, I would say. I mean, it takes a long time until you think, oh, there's something wrong, or maybe you observe them 24 hours a day and then you see a difference. So the lesions can actually develop in stages. So you can have serosal lesions that exhibit an underlying, very intense plasma cell-dominated layer of, as I said, plasma cells and B cells, and that's shown in the HE section on the right. And then on the top, you can have granulomas. And it's ages ago, but we did a study where we wanted to know about these plasma cells, and indeed, they carry antibodies against feline coronavirus. Yeah? But anyone who knows about GenPath knows that it takes at least 7 days for the first plasma cells to occur. So what we see or can see as an acute disease with an effusion can be that the animal was suffering from it for several weeks, and we see it histologically. So they can also develop in stages. So we get a bout of disease, and then it ebbs off, and then it starts again, which is why, like you see on the bottom images, we have even granulation tissue formation. It's healing, and then a new bout comes, and you see granulomas again with viral antigen. And what I found very interesting coming from this sort of experimental study, but meaning natural disease, is that some cats can survive FIP without treatment. It has been described in an experimental infection by Dutch colleagues, and the pictures at the bottom come from animals that we necropsied at the end of the study, and where we saw the leftovers, and we saw plasma cells, we saw B cells, and we saw some carrying viral antigen. And these were noted as being a bit off for a few days, these cats, and then being fine again. So they obviously managed to deal with the disease, for whichever reason, because it's still an enigma to a large extent. Some cats can confine the disease locally, like mural intestinal lesions, nowhere else, or just lymph nodes, nowhere else. It doesn't mean that they can't go on and develop FIP afterwards, but we would have seen them as necropsy cases and then find lesions, for example, only in the lymph nodes. There is more to coronavirus. You can have a coronavirus enteritis, a bit like TGE, for example. But usually you get very transient clinical signs. And we've seen a few cases where animals actually died with the coronavirus enteritis, but that turned almost, in those cases, into a hemorrhagic disease. But you find the virus where you find TGE as well, at the villus tips, in the jejunum, in the small intestine. And coming back to those animals that we had in our study, where we could prove these animals were infected, PCR was done, but they never developed FIP. What these animals showed was quite a strong T and B cell hyperplasia, big secondary follicles, macrophage proliferation in hemolymphatic tissues, but no FIP. Yeah, so they were dealing with it. They had an immune response to the virus. Let me come back to the pathotypes. And this biotype or pathotype conversion, that's, I would say, the Russian roulette for the cat, of the cat. Yeah? When does it occur? Whatever. But first, something general. Feline coronavirus infection leads to viremia. Yeah? Feline coronavirus can infect feline monocytes, and that's what it does. Feline coronavirus generally spreads from the initial site of infection, the intestine, via monocyte-associated viremia. It is generally short-lived, but it happens. And I come back to Danielle Van Moor, who was mentioned. She published that in '98. And that's a proven fact, and we can show it after infection. So healthy cats, they can be viremic for about 12 months, yeah? But cats with FIP are in the majority viremic, and we speak about a higher level. It's always monocyte-associated. And the interesting thing is that coronavirus can spread from extraintestinal sites. So there was an experimental study, IP inoculation, and the virus was shed with the feces. So it's taken up by macrophages, supposedly, and then goes. So feline coronavirus can replicate in monocytes in healthy cats. And it does it, not consistently, but it can do it. But it's higher levels of bioreplication in monocytes that we find in cats with FIP. Yeah, it's a gradual thing. And only the FIP pathotype can sustainably replicate in monocytes. The other one wanes off. And, but there was even a study where they did an oronasal infection that led to viremia, very low level, probably some macrophages picking it up, but there was no evidence of viral replication. So does this have a molecular basis? Now I come back to the 3C, 3AB, whatever. Yeah, so 3C, this non-structural protein, that is, I think it's not questioned. It's essential for sustainable replication in the intestinal epithelium and for viral infectivity, but has nothing to do with FIP development, if I word it strongly. So mutations in S, in the spike protein, are relevant for viral virulence, but more for the systemic spread and the monocyte replication than for the development of the FRP lesions themselves. And this S1-2 motif, they can lead to reduced furin cleavage, and they may facilitate viral entry into monocytes and macrophages, but it hasn't been proven. Yeah? But what we know is if you've got this FECV, enteric infection, there's a strong selection pressure that forces rapid virus evolvement. And then you have all these quasi-species. And of course, there's a fair chance that some can then sustainably replicate in monocytes. This one we can more or less forget because I mentioned it already, that we have these mutations that have been used and advertised massively by some commercial providers as the FIP diagnosis. Yes, there's a lot behind it, and it's quite likely that if you detect it and with a low CT, I mean, you have a low CT coronavirus load, you're actually dealing with FIP, but these are not the only ones. There's another one since then reported, and as far as I remember your preprint, it's a slight bit different again in feline coronavirus 23. So it's a complex situation. Most likely there are multiple mutations associated with the development of FIP. And there is one black box we haven't mentioned so far, and that's the cat. So a bit more about the virus, prevalence. It's highly prevalent and up to more than 90% in some multi-cat environments. There have been plenty of publications about that. But the morbidity is low and generally less than 10%, apart from episodic outbreaks. And the Cyprus one is one of those. There have been previous ones, but they were never as big. They were very limited locally, but there are reports on that. What are the risk factors for FIP? I mean, that comes from the general literature. Chronic shedders, the frequency of shedding, young purebred male intact cats, the season, and then there is some restricted breed predilection for FIP. That, of course, will not apply for the Cyprus outbreak. So there will be other additional risk factors. Transmission, the virus is transmitted via the fecal-oral route, and it's proven epidemiologic role of healthy carriers there. So the shedding is with the feces, and post-infection, mostly up to 2 or 3 months. But really consistent up to 2 weeks. And then follows a short immunity if it stops, but that's only for a few weeks, and then cats can become reinfected. And it's interesting, initially, while shedding, it's in the small intestine, like the other enteric coronaviruses we know, but it persists in the colon. You can't find it in the jejunum then. But it can spread from the colon back into the small intestine probably, and then they start shedding again. And there is a proportion of cats that will just persistently shed, never develop FIP potentially. And cats with FIP can shed until death, but they generally do this at a very low amount of replicating virus. What about its persistence? I mentioned the colon as the main site, but that is not the only side of persistence, because we examined a broad range of other tissues in animals that were infected, did not have a viremia, and there is in particular the mesenteric lymph nodes and the liver to mention. These are not the only ones, though, because when you have a look at this, yeah, we found it also like the left image is mesenteric lymph node with some viral antigenic macrophages, no disease, no viremia in this animal. And on the right, we found it in pulmonary intravascular macrophages. Yeah, and we also found evidence of recurrent viremic systemic spread without finding it in the intestine. So apparently viremia can develop without persistent enteric infection. There's papers about that. So what about the incubation period? That is a notoriously difficult or impossible question to answer, because after experimental infection, you will never get 100% of the cats infected, or no, you will never get 100% of the cats developing FIP. You may infect all of them, though I wouldn't be too sure, I can't remember the data. But the wet form that you then observe, it can take 2 days up to 14 days. And the dry form can take several weeks. And you can't say beforehand, this cat will develop the dry form or the wet form. So we have no clue how long it takes, really. Natural disease, no idea. So when I come back to our study, when we introduced the carriers into the group of SPF cats, we had the first clinical signs after 6 weeks. Is that the natural scenario? We don't know. So the key pathogenic features, there are a few to mention. I come to those now. The first one would be systemic infection with virulent feline coronavirus. And it was mentioned before, we have the in vivo mutation transition or internal mutation theory. That means that an FIP, the virulent form, arises de novo in an animal that is infected with feline coronavirus. Whereas the distinct circulating avirulent and virulent strains theory is the one where we have a defined virus and that circulates in the population. So what's the evidence? For the first one, the evidence would be that we have both biotypes or pathotypes in closely related pairs, so in geographically, like a phylogenetic clustering. And we have groups of cats that carry both, but very similar viruses. And evidence is, of course, that FRP is generally a sporadic disease. But then the Cyprus outbreak would follow the second theory, that we have these epidemics and that there is a virus that is basically virulent per se when it hits the new cat. And probably there is something in between, and the virulent ones will be shed at some point and infect others. It's not really clear. The other one is that we have to have an effective and sustainable coronavirus replication in monocytes. So as I said, in vitro, all coronaviruses can replicate in monocytes, feline coronaviruses, but only the ones that induce FIP will do that sustainably and spread the infection from cell to cell. There have been papers about that. In vivo is we see a monocyte-associated viremia after infection, but a higher viral replication in tissue loads with FIP. Yeah, and what the virus does as well, it induces monocyte macrophage proliferation. So basically, it provides its own target cells. And that's more intense with FIP. But I love the theory that Niels Petersen brought up in 2012 in one publication saying, what about monocytes and not intestinal epithelial cells being the site of coronavirus mutation in cats? So it doesn't end with being spread in the monocytes. The important thing, and that's what induces FIP, I think we can agree on that, is that the monocytes become activated. We need highly activated monocytes that then mediate the development of the FIP lesions, and that's the phlebitis and periphlebitis, and then the granulomatous infiltrates. So it's most likely during a phase of high-level monocyte-associated viremia with substantial viral replication that they become activated. Yeah, and how does it develop? I mean, we could see in some animals with the severe disease, and we were lucky, we found the virus in the monocytes in the blood. Yeah? They attach to endothelial cells, they form thrombi, and then emigrate. They can even form these funny thrombi attached to the endothelium. But the endothelium is not infected. Yeah? And then they emigrate, and they can form perivenous aggregates, and what I've shown you before, this big granuloma next to a vein. And we could show that the monocytes are activated. They express TNF-alpha, they express adhesion molecules like CD18. But so do the endothelial cells without being infected. MHC class II is a nice marker for activated endothelial cells in the cats, and you can see that on this vessel. So the monocytes also digest the basement membrane, so it's easy to emigrate and form these granulomatous infiltrates. And you can see here how the collagen 4 has disappeared because it's digested. So the entire sequence of events could actually be mediated by monocytes alone, provided they release sufficient cytokines to sort of set off a generalized endothelial cell activation. And of course, this is possible. So what triggers the activation? I come back, these are old papers because most recent one didn't look at this early phase. So we've isolated monocytes, alveolar macrophages, they were infected in vitro, and in the early phase, during the first 6 to 12 hours, we saw an upregulation of cytokines, pro-inflammatory cytokines, TNF-alpha, IL-1. Now, if you wait a bit longer and let the virus replicate, you see VEGF going up. Okay, that's interesting when you look at when you consider that you're looking at a phlebitis. And then they waited even longer, and they got TNF-alpha, VEGF, but then come some colony-stimulating factors, GCSF, MCSF, and these are the ones that will set off the monocyte macrophage replication. So more target cells, great. You know, it's like a spiral that then, I think, sets off. So I come back to a scenario of feline coronavirus infection turning into FIP. So the one, it's a few clicks now, you've got an enteric infection, right? You have infection of monocytes, fine. The virus is, we have viremia and infects tissue macrophages. But then you have, according to the theory, you've got the mutation in the enterocytes. And from there, you get viremia, replication of the virus, activation, and you eventually get the phlebitis. Now, what if monocytes and macrophages were indeed the site of feline coronavirus mutation? The option would be that we have a positive selection for macrophage tropism and progressive adaptation to replication in monocytes and macrophages. There is some evidence of it that we have apparent viral clearance from the blood and intestine that does not prevent the recurring viremia and FIP. There is experimental evidence that there is at least a viral transformation at the enterocyte, macrophage, and then monocyte interface. So in principle, that would mean that FIP could develop at any time post-initial infection, because the viremia, monocyte-associated, will develop anyway, even low level. So we have these persistently infected animals. We have a high mutation rate of the virus. Why not? And now my other scenario, lots of clicks. So you have the replication in enterocytes. It goes into monocytes. The monocytes, viremic, go into the tissue. Pulmonary intravascular macrophages, Kupffer cells in the liver, yeah. Then the cells in the mesenteric lymph nodes. We have macrophages in tissues. We have fully differentiated tissue macrophages, yeah. Okay, now think it happens in the monocytes themselves. So yes, that's easy, can induce the vasculitis, but it could also lead to recurrent viremia from the tissue macrophages, and then the mutation in the monocyte, or we could have the mutation even like in Kupffer cells. And we could have the same scenario. We can't, at the moment, we can't tell. So there have been studies on the cytokine levels and the transcription in association with FIP. And that's very valuable information. You can see upregulation of TNF-alpha, but you can also see most cytokines go down when you actually see the patient and it's about to die. Yeah? So there is a lot of data, but it depends really on the phase of the disease, what you can detect at high levels. But again, it might be that the monocytes can be directly responsible for everything. So maybe fulminant monocyte activation, only as brief bouts, set off the vasculitis, and then we're looking at a self-sustained granulomatous process. It's not unlikely. So just 2 or 3 words about the immune system. Yeah, sorry. Oh, I'm sorry. I had 30.
Yeah, but that includes questions.
You don't have any questions, do you? No. Do you want me to stop or finish and not have questions? I can't speak faster. Sorry about that. Now you stopped me on track. Julian. So we have high antibody titers, but they do often not eliminate the virus or the infected cells. We have rapid internalization, and ADE was mentioned. That may also play a role. And we have a lack of viral antigen expression on the surface of the infected monocytes, and that might inhibit cell lysis. So feline coronavirus, to some extent, hides from the immune system. So maybe that this leads to viral persistence options. Yeah? And when we have FIP, what happens is actually that we have a massive B and T cell depletion, and these animals show lymphopenia. So that happens at some point, and it has been speculated that this comes from the high bouts of TNF-alpha expression at some point. And then we see the massive macrophage proliferation. And it's quite interesting. We did the transcriptomic studies on the mesenteric lymph nodes. You have infected cats and no FIP. There you can see in the lymph nodes you have a direct viral effect by TLR9 and STAT2 upregulation. But in the spleen, for example, IL-10 gets up, so probably it limits the inflammatory response. When you look at the lymph nodes in FIP, we do not only have this first innate immune defense and a direct viral effect, but we also have this inflammatory pathway activation. So something then doesn't stop the inflammatory response when the animals develop FIP. And again, it might be that it's the macrophages that do the job, I'm sure. But when you have a cat with FIP, cytokines are also produced in other organs. We found it in hepatocytes. We even found it in the myocardium. They express or transcribe cytokines. So that will all work towards the systemic inflammatory response. So we have probably an acute phase response driving it. But one more thing, and this is my last slide. A few facts on the susceptibility of cats to FIP. Individual cats can be entirely resistant to Feline coronavirus infection, and we don't know why. The monocytes of individual cats can be completely resistant to coronavirus or to the less virulent feline coronavirus alone, so not much will happen. The monocytes of some cats are more prone to productive coronavirus infection than those of others, and also you can have monocytes and you can infect them today, but you may not be able to do that in 4 weeks' time. That happened to me with one of my in vitro experiments. Yeah? Cats can be resistant to FIP, they have a strong CMI, have high interferon levels, but that's work done in Bristol. Interferon SNPs are not discriminatory. No effect on the FLA, no effect of blood type on the antibody response. So that's it. And just a few people to thank. And I'm very sad to say that Emeritus Professor Hans Lutz just passed away a couple of weeks ago, so, but all others are still alive and kicking. And that's different places I worked. And you see Alex Melbourne, who was a PhD student of mine, who's now a pathologist in Edinburgh, and Severin Tasker as well. We did quite a bit of work together. So I won't have my take-home message. Thank you. I know there are no questions, so I stop.